In this video, we will talk about gene regulatory networks. We know that in the process of transcription, RNA polymerase makes copies of DNA into an RNA sequence, which is used later during the process of translation to make proteins. However, genes can be turned on or off in the presence of certain gene regulatory proteins. For instance, if a repressive protein binds to the DNA which blocks the binding site for RNA polymerase, then the genes will not be expressed. On the other hand, sometimes the RNA polymerase cannot bind to the DNA by itself and instead needs the help of an activator protein which allows the RNA polymerase to bind and promote expression of the genes. Sometimes the genes can encode for regulatory proteins themselves, which can cause a chain reaction of events in which expression of one gene promotes or inhibits the expression of another gene, which in turn can promote or inhibit the expression of another gene, and so on. This interaction of regulators that determine the expression of genes in DNA is part of the gene regulatory network. As an example, let's say we have a series of genes that encode for characters in the movie Star Wars Episode 4, A New Hope. The Darth Vader gene inhibits the transcription of the Princess Leia gene. The Princess Leia gene promotes transcription of the Death Star plans gene. The Death Star plans gene stimulates transcription of the Luke Skywalker gene. And finally, the expression of the Luke Skywalker gene inhibits the expression of the Death Star gene, and the Death Star is destroyed. Here is a visual representation of this developmental network, where you have Darth Vader suppressing Princess Leia, who stimulates the expression of the Death Star plans, which stimulates the expression of Luke Skywalker, who needs those Death Star plans to destroy the Death Star. But what if there was a mutation in the Darth Vader gene? Well, Darth Vader would no longer be able to block Princess Leia from promoting the expression of the Death Star plans, which would be used to express Luke Skywalker, who would then be able to destroy the Death Star. If the gene for the Death Star plans was mutated, then Darth Vader would still be able to block Princess Leia, which doesn't really matter because the Death Star plan gene is mutated and can't be expressed anyways, which means that Luke Skywalker cannot be expressed, and so he's unable to block the Death Star. Now, if we had a double mutant in which both Luke Skywalker and Princess Leia are mutants, then Darth Vader would essentially serve no purpose since the Death Star plans cannot be expressed without Princess Leia, which really doesn't change much since the Death Star cannot be blocked by Luke Skywalker anyways, and so the Death Star is not destroyed. What if we had another double mutant in which Darth Vader and the Death Star plans were mutated? Well, without Darth Vader, the expression of Princess Leia is not inhibited. However, she is unable to stimulate the expression of the Death Star plans since it is a mutant, which means Luke Skywalker is not expressed and the Death Star is not destroyed. So as you can see from this example, the interactions of different characters in a movie determine the outcome of the plot, much like how different genes in a genome can interact with each other to determine the resulting phenotype. Do 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 do. This song is copyrighted, so I can't use it. Thank you.